talk. I think this might be a more appropriate title because I'm really going to talk most of the time about suspension feeding. And then at the end, I'll come back and talk a little bit about swimming. We have a couple projects, one looking at Mako swimming and one looking at acceleration in lampreys. So I'll just start out with a little bit of background on suspension feeding. This work was done at the University of Maine. I was advised by Pete Umars and Damian Brady, who helped out a lot on this work, and I also had some help from some undergraduates, Ian Jones and, well, looks like the other one didn't show up, Tyler Carrier. He helped too. So this is the study organism. This is my Arenaria, the soft-shell clam. They're pretty common up in Maine. They're an infaunal marine suspension feeder. And suspension feeding is important for a variety of processes. Essentially, benthic suspension feeders link benthic and pelagic ecosystems. So they take carbon and nutrients that are in dilute concentrations in the form of phytoplankton and particulate organic matter, and they concentrate them by increasing their own biomass and by producing biodeposits, which sink quickly out of the water column. And so this can be particularly important in eutrophic environments that are subject to large blooms. They can help, having a healthy suspension feeder population can help control some of these blooms and help limit some of the negative effects such as eutrophic, anoxia. So for all these reasons, it's important to quantify suspension feeding, and people have been trying to quantify suspension feeding for a long time using various methods. The most common methods are the so-called indirect methods, which measure a clearance rate. And so basically the idea is you take a tank and you put in your suspension feeding organism, and you add some particles, such as phytoplankton or some kind of organic or inorganic particles, and then over time the suspension feeder will feed and decrease the concentration of these particles, and so by taking the concentration over time, we get some measure of suspension feeding activity in the form of a clearance rate, which is the volume of water that's cleared of particles per time. There's also another quantity that's important, and that's the pumping rate. And so that's Q here, and that is just the volume of water pumped by the animal, regardless of whether or not it contains particles. And the two are related by this retention efficiency. So if there's a certain type of particle and a certain size of particle, and a clam retains 50% of that particle on its gills, then the clearance rate will be half of the pumping rate. And so these methods are very useful, and they've given us a lot of interesting results. They're good for comparing species, looking at the effects of temperature, but they don't apply very well to the field, and it's difficult to compare between experiments because they're subject to wall effects, other experimental artifacts. So there are another set of methods called direct methods, which measure pumping rate directly. And so this is a very early example from 1926, published in Science. The author of this paper essentially took an oyster and jammed a rubber tube into it and to look at the water flow through that tube as a way of measuring pumping rate. So obviously that has some limitations. That's probably not going to be the same rate as you see in the field. But we've come a long way since then, and we have some newer methods, some of which are non-invasive, that produce similar numbers. So going back to my Arenaria, now this is a view from above. I'll just talk a little bit about how they feed. So my Arenaria has a pair of fused siphons, an inhalant and an exhalant. The inhalant is larger, and so the animal draws water in through its inhalant siphon and then that water passes over the gills, and the gills extract particles and oxygen, and the depleted water returns out through the exhalant siphon in the form of jet flow. And so jet flows are pretty well understood from a fluid dynamic perspective. They've been very well studied. They're important in aerospace, for example, but there's surprisingly little work done on inhalant flows. So we focus here on the inhalant flows partly because of their importance for fluid dynamics, but also because they're very important for the animal, because these flows determine what resources the animal has access to. So be they chemical cues or food or oxygen, this flow essentially connects the animal with the rest of the ocean. So there's a couple of numbers that are important for these flows. This is just a simplified inhalant siphon flow. We're just looking at streamlines going into a siphon. The most important number is the Reynolds number. So this is a dimensionless number. It relates. Am I missing something?
Oh no, I'm just nodding to my students. I said, you know about it.
Oh, okay. Good. Excellent. So you know that the Reynolds number relates excuse me. Now I forgot. So the Reynolds number is found by multiplying the density by the mean velocity and the siphon diameter in this case, and dividing by dynamic viscosity. And so this number is uniquely defines this flow. So if you have half the velocity, twice the diameter, you'll have a very similar flow. Another important number is that pumping rate that I mentioned before, and one way to find it is using Poiseuille's law, which is describes flow through a pipe, or in this case a siphon. So it's found, it's related to the dynamic viscosity, the pressure gradients, so the pressure gradient along the siphon, so pressure, the fluid flows from high to low pressure, so the pressure decreases along the length of the siphon, and the siphon inner diameter. And so you can see this is a fourth power here, so this term is going to be very important, and a little bit later on I'm going to be coming back to why this is such an important term. Okay, so that's most of the equations. This is the method we used. It's called particle image velocimetry, or PIV. It's a way of measuring fluid flows, and basically the way it works, you take a tank filled with water and you seed it with some kind of a particle that will very closely match the flow. Follow the flow, and then take a series of images, and then based on the movement of particles from image to image, you can reconstruct the flow with very high spatial and temporal resolution. So this is the setup we used. It has a laser, a tank, and a pair of cameras. The laser has optics to bring it out into a sheet, because we want to focus just on one plane. And we film with a pair of cameras in this case in order to get 3 velocity components in this plane. So here's an example of an image from a PIV experiment. This is my air and area again. Now we're looking at the inhalant siphon, and the exhalant is directly behind it in this image out of the plane. So taking a pair of these images, we can calculate a velocity field that looks like this, where the warmer colors here are higher velocities. And so you see a lot of vectors pointing in towards the siphon, which makes sense because this is an inhalant siphon. Over here you see some vectors pointing in the other direction, and this is because of the influence of the exhalant siphon. Remember, the exhalant is behind the inhalant here, but the exhalant siphon is a jet flow, and it produces much higher velocities than the inhalant flow, and entrains water into it. So we see the influence of it here, even though we're not focusing on it. So now this is just time average over 40 seconds of these velocity fields. And in order to compare different animals, we take a transect. So we'll start at the center of the inhalant siphon, and then we'll move out along the axis away from the siphon. And one thing you might notice here is that the velocities are actually very low right by the siphon. And this is a convergent flow, and we know that the velocity should continue to increase as we approach the siphon because the water is going through a smaller and smaller area. The reason that we see such a low velocity here is because of the laser. So the laser reflects off of the siphon and it washes out the image, and we can't get accurate velocities in this region. So this gives us a velocity profile that looks something like this, where if you start on the right side of this plot, you're looking at very far away from the siphon, and then as we move in along that axis, we see an increasing velocity until we get to that region where we can't accurately measure the velocity, and then the velocity drops off again. So this is good, but unfortunately we really need the velocity right at that siphon because we can use it to calculate the pumping rate, which is what we're really interested in. This is the suspension feeding rate. So, and so we can find that by multiplying the area by the velocity at the siphon inlet. So one approach to getting that velocity might be to fit a function, in this case an exponential function, and just extrapolate. And this approach has actually been used before, but we actually have a more accurate method that I'm about to show you. This is what we use. So this is an example from computational fluid dynamics, or CFD model. We take a geometry, in this case the inhalant siphon, and a set of boundary conditions, and based on that we can recalculate the entire flow. And so we get the flow right up to the siphon inlet, which is really what we're after. And so in just the same way as we did with the PIV, we take a transect just right along the axis. We run this over a range of Reynolds numbers, and we get a set of profiles. And so by fitting these to the PIV data, we can choose the best one, and we can calculate the Reynolds number. So if we add this onto our earlier plot, we can see that this is almost an order of magnitude higher. So that initial approach of using the exponential function really wasn't giving us the real picture. And this higher velocity also translates into a higher pumping rate. The two are linearly related, so we have also an order of magnitude higher pumping rate. So we did this for 9 animals, shown here. And we related this to size of the animal, in this case in shell length. We can also use this to calculate the pumping rate. In this case, we found a linear relationship, and this is similar to what was seen for clearance rate experiments. We also looked at a few other species. This is Mercenaria mercenaria, the hardshell clam, and Siona intestinalis. It's a solitary tunicate. And so we got a range of Reynolds numbers and pumping rates for these animals. And you can see there's quite a range here from Myoarinaria, which has pumping rates of 1.7 to 7.4 liters per hour, where Siona only has pumping rates between 0.1 and 1 liter per hour. So the reason we do this is we're really interested in getting more accurate suspension feeding values in order to feed into ecosystem models, for example, various reasons to quantify suspension feeding. But also these models, I didn't have a chance to get into that much, but once we know what the entire flow, we can do some interesting things with the model. For example, this is an inhalant siphon with based on realistic siphon tentacles of a species of clam, and we're looking at the origin of all the water that's sampled over time by this clam. So anything in that green blob it has access to, and anything outside of that green blob it doesn't. So now that we have this method in hand, we wanted to look at some other problems, and one of these is juvenile suspension feeding. So here's my area again, but these are about 2 months post-settlement out of the water column. They have about 2 millimeter long shells, and this is a very important life stage for these animals. They have very high mortality rates, sometimes up to 90%, and they're growing very rapidly. So their suspension feeding rate is likely to be very important, even more so than the adults perhaps. And so in order to, we did something similar with the juveniles using a technique called micro-PIV. And this was done with Husho Zhang at Woods Hole Oceanographic Institution. This is his micro-PIV system. It has a high-speed camera, lens, which is a long working distance microscope objective, a tank to hold the animal, and a light source, which is a collimated LED beam. And so here's the animal in the setup. It's a little small, so he's right there. One of the advantages of this method over traditional PIV is it gets really nice clear pictures because you don't have to rely on using a laser. In order to get the sheet that we need, we're actually relying on the microscope objective and its narrow depth of field. So just to orient you, this is the inhalant siphon again. This is the exhalant. The inhalant siphon is ringed with tentacles similar to the adult. The exhalant has this sort of extension on it that is not found in the adults, and that's something I'm going to come back to later on. So if we take a stack of these images and combine them, we get path lines shown here, and this will allow us to look at some of the features of the flow. So first, here's the inhalant flow. You can see the water converging in towards the inhalant siphon. Here's the exhalant jet, and you can see the water accelerating. You see both the water coming out of the exhalant siphon as well as water being entrained into it, and as it accelerates, it contracts. And there's a region here where the flow separates. So part of the flow goes into the inhalant siphon, and then part is entrained into the exhalant flow. And there are also these two big high vorticity regions, one positive and one negative, and these are likely slices of a vortex ring that surrounds the exhalant siphon. And so here's a velocity magnitude, velocity field, similar to what we saw for the adults. And just as we did with the adults, we do the same thing with drawing a transect, finding the Reynolds number, and looking at a range of shell lengths. So that relationship is shown here. And again, we calculate a pumping rate. And one thing to note here is, remember for the adults, the pumping rate scaled linearly with shell length. Well, here we have an exponent of 6.5, so clearly there's something very different going on here. And so this becomes even more clear if you put the two together on a log-log plot. So how do we explain this? Well, we think that this comes back to Poiseuille's law. We think that siphon diameter is the major limiting factor in determining pumping rate for these juveniles. And that comes back to that fourth power. So if we look at pumping rate in terms of siphon diameter, we see a similar exponent of 4.7. So it seems that these juvenile clams are pumping as much as they can with the size of the siphon that they have. So as they continue to grow, they very quickly increase their pumping rate. Whereas the adults, their siphon is probably already has a large enough diameter, so they're limited by something else. And in the literature, one suggestion is that it's the area of the gills that limits pumping rate. And we also think that this might mean that there's a limit on the size of suspension feeders. lower limit where if the siphon is too small, that really isn't effective to suspension feed. And there's actually observed behavior known as pedal feeding, where they feed with the feet and they sweep water in towards their mouths when they're much smaller. So going back to this exhalant siphon extension, this structure has not really been well described in the literature, and there's not really a good explanation for its function. Based on what we see here, we think that the purpose is to redirect the flow, to get that exhalant flow away from the inhalant flow so that they don't refilter water and waste energy pumping the same water over and over again. The adults, they have a mechanism for dealing with that, and that's just to produce a very powerful jet. So these jets are on the order of 1,000 Reynolds number, and they carry the exhalant flow very far away from the inhalant, whereas the juveniles, they don't have this luxury, so they have to have another means to deal with the problem of refiltration. And we also have models showing that the refiltration is almost completely eliminated if you add this extension, and if you don't have the extension, refiltration happens almost right away. This is some of the first measurements of suspension feeding rates for juveniles. They've been a little bit neglected. They're in between larvae and the adults, both of which have received some attention. And I think one reason that these rates have not been measured before is because of the clearance rate method doesn't really work with them. They're just too small, and they produce such slow flows. So this is sort of the first time that we've been able to see what they're doing. So now I'm going to completely change topics and talk about swimming. I'll talk a little bit about swimming in Mako sharks. So this is the Shortfin Mako. They're the fastest swimming sharks. They also swim over great distances. They can migrate up to a couple thousand kilometers or more. So drag is likely to be very important for these animals, and minimizing drag. So this project is a collaboration with my advisor at USF, Brad Gemmel, and Phil Moda, also at USF, and Amy Lang at University of Alabama. And the idea is to look at the flow over shark scales to see if there's some mechanism that they use to reduce drag. And this is a picture of shark scales. You can see they're pretty small, about 0.1 millimeters long. And so in this picture, the shark would be swimming from the right to the left, meaning the flow direction is to the right in this picture. But remember, they're swimming very, very fast, and so this flow is turbulent. So there are flow reversals, there are turbulent eddies and other features that go in the opposite direction, and these actually can pop these scales up. And the idea is that when they do, they prevent some of these features from forming, and this helps to reduce drag. So we filmed this using micro PAV again, and in this case, the scales, if you'll excuse the pun, are very different though, because we're looking at meters per second. And so we had to film this at 50,400 frames per second. So we're going to see this slowed way down to about 30 frames per second. So you can see the flow is, for the most part, from the left to the right, but then occasionally we get these turbulent eddies and backflows that come back in the other direction, and they pop up the scales, and the scales pop up and down in the flow. So we wanted to look at what sorts of flows cause this behavior in order to better understand how these scales are functioning. Yes?
Is it just like a piece of skin that you guys have in like the tank? Or?
Yeah, so this is just skin. Okay, this slide's a little complicated, so I'm going to sort of orient you to it. The top are two velocity fields. One is when the scale first moves, so when you first start to see the scale pop up, and then the second one on the right is when the scale reaches its maximum angle. And the plot below is velocities over time, and so the first dashed line refers to the first scale movement, and the second refers to the maximum angle. And we're looking at the components of velocity here. So in orange is the horizontal or x velocity, in blue is the vertical velocity, and in black is the velocity magnitude. But if you don't want to look at that, there's also arrows here that show you the direction of the flow. This is a backward jet. So this is the first case we looked at. This is the opposite direction from the way the flow would be going if the shark was swimming forward. The reason we did this is we wanted to see the maximum that these scales would pop up. So if you follow this timeline, you see that once the velocity reaches a certain magnitude, the scale pops up. The scale's pretty much almost entirely horizontal. So it's really just flow going over the scales and pushing them up from right to left. The next case we looked at is a more realistic case, which is a forward jet. So the water is going in the direction it would be for forward swimming. And we put in a fence upstream of the scales to get some of these features, these sort of turbulent features that we would see in a naturally swimming shark. And so first thing you might notice here is the velocities are much lower, and we don't actually see that threshold effect here. What we see is the scale doesn't actually move until the flow switches direction. So it starts out sort of down and to the right, and then when it flips direction to the left, it comes under the scales and it sweeps them up. And then, so that's where this arrow changes direction. We see that the flow comes in underneath the scale and it pushes them up. And then at the maximum angle, the flow is almost completely horizontal here. And when the flow switches back the other direction, the scales pop back down. But we also did the same thing, and we found something completely different. And in this case, the flow is entirely from left to right until almost the very end. So that doesn't really make a lot of sense. You would think that this flow would just be holding the scales down. So what we think is important here is this vortex here, so this feature here. So we looked at that a little bit more closely to see if this vortex was having some effect on pulling up the scales. So here we take the vortex and we draw a transect starting in the center, and we look at the velocity components. We can also calculate vorticity, so we see a pretty high vorticity region here, which makes sense. Another thing we can do is we have some code developed by John DeBerry at Stanford that allows us to actually calculate pressure from the velocity field, which is really nice. So we can see that this actually creates a very low pressure region near the center of this vortex, and we think that this low pressure is actually pulling up the scale as opposed to the other case when it was being pushed up from underneath. So it gives us a better idea of what's happening with these scales. So the reason to study this is to look at drag reduction. Amy Lang's lab actually has some funding from DOD, and they're interested in coming up with some kind of structures to put on helicopter propellers to reduce drag, or other similar applications. So I'm just going to have a very brief slide on lamprey swimming. We looked at escape responses in sea lampreys. And we found that they actually push off with their head, and they develop this high pressure region next to their head, and this gives them, allows them to produce thrust about 0.2 seconds earlier than thrust developed by the, than the thrust that the tail produces. So this actually gives them a bit of a head start, which can be very important in the case of an escape reaction when they're trying to escape from a predator. So I'm really not going to get into this. I'm mostly just trying to plug my talk at Ocean Sciences. I think a lot of people, you'll probably be there. It's on Friday right before lunch. So I worked with a lot of people on these projects. University of Maine, Pete Umars and Damian Brady were my advisors. Again, I had help from Ian Jones and Tyler Carrier on the PIV. This work was a collaboration with John Grimaldi's lab at UC Boulder, and we had funding from the NSF. At USF, my advisor is Brad Gemmel, and I've been working with Phil Moda. Amy Lang's lab at University of Alabama first started with this shark scale studies, and John DeBerry used that pressure code that has allowed me to do a lot of this work. And again, NSF funded this work. So